The spotted wing drosophila is a threat to fruit growers. This fruit fly made its way from Southeast Asia in 2008, likely hitching a ride on fruit shipments. First detected in California raspberry fields, the insect rapidly spread to other states, causing hundreds of millions of dollars in damage each year.
It's here now. Wherever you see berries being grown, you find the spotted wing drosophila infesting fruit.
Scientist Stephanie Gamez is an expert on this invasive fruit fly. So, when she was asked to join the St. Louis-based biotech company Agragene as the director of R&D, Gamez took her very own spotted wing drosophila from San Diego on a road trip to St. Louis.
Thousands, over 10,000 flies packed up the lab.
Along with her family, including the cats. And in the tubes, in the tubs, the gene-edited spotted wing drosophila.
My precious spotted wing drosophila lines.
The troops headed to St. Louis are the Hope, an army of gene-edited sterile males that may one day stop the threat.
That were completely sterile, and those were precious. That was the product that would make the company in St. Louis.
The idea is that if they were to be released into the environment, the sterilized males would mate with wild females, resulting in a fertility dead end. Gamez and the Agragene team use a precision-guided sterile insect technique.
A new technology that's built off an old technology. So sterile insect technique's been around for over 50, 60 years. It's been used by the government to basically decrease the population of invasive species. What we've done is reinvented it. Basically, we're using CRISPR-Cas9 technology to do editing of flies' DNA in order to make it sterile and also not to produce females.
The product that we're trying to sell is the edit that makes the male sterile. The precision sterile insect technique is a CRISPR-based system. CRISPR technology is using molecular scissors to target and cut DNA at specific target sites. And so if we put that in the fruit fly or in the spotted wing drosophila in our case, and we generate these flies that have these tools, these molecular scissors, and we mate them together, we make instantly sterile insect males. And the idea is if we produce enough of these sterile males and then release them in the field, see less fruit flies in the field and also less infestation in the crop.
We have to work with the government, with the USDA. We also have to work with the EPA to get permission to do broader testing. But we need to get the okay from them because it is something new that we're releasing out into the fields.
Gamez led the research team on the transition from the lab into contained field testing, releasing the gene-edited flies on berry farms.
We were able to do some work in California in raspberries, blackberries, and blueberries, and then to do some work in Oregon on cherries and blueberries.
These sterile males, they just harbor these gene edits and they behave like normal wild males. Wild females, when she comes across our sterile males, she doesn't know they're sterile, right? Because there's no, peacock feathers or anything saying, hey, I'm sterile. So once that sterile male mates with her, she feels mated and there's these changes happening inside her that increases egg production because she thinks she's mated. But in reality, when she lays eggs, they're unfertilized.
Precision genetic methods are considered efficient and environmentally friendly. Currently, growers rely on pesticides to protect crops from the fruit flies, but pesticides kill helpful insects.
Important insects like pollinators, like bees, those are critical for like blueberries, small fruit.
Gamez believes the hope that accompanied her on her move to St. Louis may one day save the day with her pest and others.
So this technology is a platform technology, meaning like you could apply it to almost any insect. As long as you could insert it in those bugs and you get sterile males, you're good.